Hello, this is Team iGEM NCKU Tainan. We're the Taiwan NCKU team for the iGEM competition this year. The competition is about solving real-life problems with synthetic biology. Our team is composed of 3 groups, wet, dry, and HP. This audiobook project is made by our human practice team, whose job is to share our topic with the public and let people know more about synthetic biology. We will have 2 audiobook episodes, each of which will contain 3 different kinds of topics related to synthetic biology. Now, let's get started with our first audiobook.
National DNA Day. Do you know what National DNA Day is? It is celebrated on April 25th, which commemorates the discovery of DNA's double helix by James Watson and Francis Crick in 1953, and the successful completion of the Human Genome Project in 2003. Due to James and Francis' research, they discovered that DNA consists of 2 polynucleotide chains and 4 types of nucleotide subunits. The nucleotide subunits are glued in pairs with each other by the hydrogen bonds. So, what exactly is Human Genome Project? It is a worldwide project held by National Human Genome Research Institute since 1989, aiming to determine the base pairs that make up human DNA. Up to 2003, more than 92% of gene sequences have been found. The database is now available on the internet and can be searched by everyone. It has been almost 2 decades since the completion of the Human Genome Project. We are a lucky generation who can accurately read our genome through fast and efficient genetic testing instruments, and compared it with the prevalent disease database of many years' research to understand more clearly the individualized disease. In this way, effective disease prevention measures and targeted treatment methods are formulated. Besides celebrating, the goal of National DNA Day is to offer students, teachers, and the public an opportunity to learn about the latest advantages in genomic research and explore how those advances impact our lives.
CRISPR beef cattle get FDA green light. In March 2022, the U.S. Food and Drug Administration (FDA) declared that products from 2 CRISPR-edited beef cattle pose no risk to people, animals, the food supplies, and the environment. The cattle, which is developed by a biotechnology company from the U.S. called Aceligen, have a slick hair phenotype that not only allows the animals to better withstand tropical heat, but also allows the cattle to grow faster and stronger. What is CRISPR? Its full name is CRISPRCas System. It is a fast and strong genome editing tool that can be applied to bacteria, plants, animals, and even humans. It was first discovered in E. coli as an adapted immune system to avoid foreign DNA from viruses. Its mechanism could be thought of as a cut-and-paste process. The guide DNA would recognize the specific sequence called the PAN and bind to the following complementary DNA region. Then, the Cas enzyme can be guided to the target position and act like a scissor to cut the DNA region precisely. Researchers can modify the genome fast and precisely by designing the guide RNA. As a promising gene editing tool, the CRISPR-Cas system can serve as a strong tool to solve problems like cancer, malaria, and of course, the food crisis in the future. These genome-edited animals are called IgA animals. The full name of IgA is International Genomic Alternation, a general name for many kinds of genome editing such as CRISPRs, zinc finger nuclease, talents, and ODM. To reflect the newest use of biotechnology, in 2008, the US FDA first issued draft guidance, which is called GFA 187. It's a guideline now used to deal with the IgA animals. Under GFA 187, there are several products that had passed a low-risk determination for enforcement discretion. For example, Aqua Advantage salmon was engineered to reach growth markers more rapidly than its non-genome edited salmon counterpart. In 2010, a public hearing on the labeling of food made from Aqua Advantage salmon was held, and it is the earliest IgA product. Another example is that in 2020, FDA approved an IgA product called GalSafe Pigs, which can be used for food or human therapeutics. It is the first IgA in an animal that the FDA has approved for both human food consumption and as a source for potential therapeutic uses.
Cosmetics. Do you know that even cosmetics can be produced in a biotech way? Squalene is an organic compound used for skincare products and vaccine adjuvants. In the past, squalene was extracted from the livers of deep-sea sharks. However, only 7 grams of squalene can be retrieved from a single shark, while the demand from the market is over 2,000 tons per year. Hence, the hunting process can cause great damage to marine biodiversity. To solve this problem, a biotech company called Emrys is now offering a sustainable alternative. They use farnesene, a genetically modified yeast which is produced from the fermentation of sugarcane sugars. Then it can be dimerized to squalene under some specific chemical converts. With this new symbiotic method, our marine ecosystem has been preserved. Aside from squalene, there are other raw materials for skincare products that are produced by a symbiotic way, such as collagen, elastin, and hyaluronic acid. Collagen and elastin are used to improve skin elasticity. They once came from the bones and skins of boiled animal bodies. Now, there are lots of alternatives to production. One of them is by engineering ribosomal pathways and amino acid secretion machinery in microbes to optimize protein production. As for hyaluronic acid, it is a valuable ingredient for cosmetic and medical use. For decades, it was sourced from rooster combs. Now, the most common way is using engineered Bacillus subtilis, which expresses the Has-A gene from a kind of cocci that encodes the enzyme hyaluronan synthase, resulting in the production of hyaluronic acid.